Right. So we want to continue on with our look at the various pieces or parts of trace evidence. And today's discussion will be on pollen and spores. Now, when you look at evidence that's going to be collected from basically plant-based material, uh, rarely would you find this as a standalone piece of evidence. Oftentimes, this is going to be used to confirm or verify certain movements or activities of an individual, or maybe even locations that the individual had been. Uh, but rarely would this be the kind of the smoking gun, if you will, that would be used to provide the sole base for incrimination of a particular individual. So you want to kind of keep that in mind, obviously, as we look at this particular section. But when you look at it, it's known as palynology, looking at the information that you're going to get from um, pollen and spore production or pollen and spore evidence, specifically as it relates to those reproductive structures that are going to be produced and released by various types of plant material. Now, again, because of the size of pollen and spores, they're exceptionally small. Many of them are going to be microscopic or nearly microscopic, and because of their durability. So again, these structures have to be made in such a way that they're going to offer the best likelihood of being able to withstand a wide range of environmental conditions to fulfill their ultimate goal. Their ultimate goal, obviously, is going to be used to perpetuate the species of those particular organisms. So these structures are made to be very, very durable. Now, the benefit to us as human beings is that obviously, from a forensic standpoint, that allows us to be able to study these things for long periods of time, and they can be available for very, very long periods of time. So from a forensic standpoint, again, it's going to allow us to be able to know whether or not individuals have been in certain locations at certain times of the year, because obviously plants aren't reproducing year round. There are going to be different times that they're going to be more active in the amount of pollen that they will produce, or even the type of pollen that is produced by other plants in the same area. So it's not that all plants reproduce at exactly the same time. And so again, the prevalence or the amount of pollen that you might find in a certain location at a certain time can also be used to indicate maybe a time of year that an individual visited a particular location, or something along those lines. So that ends up being obviously very, very useful. Um, as I mentioned, they are very durable. So the textbook has a story where they're talking about a mass grave had been uncovered in 1994, contained 32 male bodies, and so when they found this mass grave, there was a little question, or a question as to whether the grave was that of um, a number of executed individuals by the Nazi secret police in 1945, or whether they were Soviet soldiers that had been killed by the Soviet secret police um, in the early 1950s. And so they just based on material that was there wasn't anything that was apparent that would identify them as maybe being individuals that had been executed by the Gestapo, or whether they were individuals that had been executed by the Soviet secret police. However, further inspection allowed them to find um, some pollen and spore residue in the nasal passages of the skeletal remains. And so, as they started looking at that, what they found were some pollen grains from lime trees and rye. And so, what they found is that all of these plants release pollen in June and July, and so they determined then that the bodies belonged to those of Soviet soldiers that the Soviet secret police had killed in the summer of 1953, and not people killed by the Gestapo during the spring. So again, you can kind of see how the durability of these particular seed plants benefits forensic scientists, but obviously a primary use is going to be to benefit the plants and allow them to be able to be be fertilized or pollinated, I guess I should say. So both are reproductive structures, microscopic and resistant to decay, resistant to drying out, resistant to lots of different things. That's going to allow them to persist and survive, like I said, a wide range of environmental conditions. That's a beneficial thing. So when we look at pollen production, um, those plants that release pollen and really even spores to that degree are going to be able to generate what's called a pollen fingerprint. And so what this simply is, it's a description of the amount of pollen and the types of pollen that are going to be found in a particular location at a particular time of year. So obviously you would not anticipate it being the same in August as what it is in, I don't know, April or May, right? If we're talking this area, obviously our a lot of plants in our particular location in Ohio, you'd find a lot of pollen grains uh, in really large quantities in April, May, maybe even early June, as we have a lot of our plants that are kind of really ramping up in terms of reproduction. And so you would anticipate those numbers being a lot higher in late spring, early summer, as opposed to late summer and even early fall. Though you would have some that would be reproducing, uh, but again, the types of pollen and number of pollen grains would change based on what those might be. Okay. So when you're looking at your non-seed plants, those plants that don't reproduce using seeds, these are going to be our gymnosperms. Going back to some old biology stuff here, those are going to be your naked plants, right? In terms of their reproduction, so they don't use pollen; they're going to use spores. So these would be your ferns and mosses primarily. There's a few other groups of plants that you would find in this category as well, but a lot of these are going to be your cone type of plants. So pine cones, cycads, ginkgos, those types of plants are going to use spores in place of or um, in lieu of pollen grains, or in addition to those pollen grains, I guess I should say. All right, so again, very useful. And then you've got your seed plants, your angiosperms. They're going to use pollen to produce those flowering plants. Um, so yeah, so there's that. So again, we can use that obviously to help identify the different organisms that are going to be present. When you look at how these plants will pollinate, those that use pollen, um, there are some plants that are self-pollinating plants, like garden peas are a good example of that. And your self-pollinating plants, because of the efficiency, because of the accuracy, excuse me, accuracy with which the pollen reaches the part of the flower that needs to be fertilized. Because of that accuracy, they don't need to generate the same amount of pollen as those plants that are going to use cross pollination, meaning you're relying either on insects or wind or water or something like that to get your pollen grains to the female portions of the flower. And so because of that, there's going to be limited value associated, excuse me, with self-pollinated plants as opposed to cross-pollinated plants. Though you could also argue that cross-pollinated plants, because of the sheer volume of pollen that they're going to produce, is going to be limited or have limited forensic value as well, because it is so prolific, or there is such large amounts of pollen from these things. Again, not necessarily always super useful from a forensic standpoint. So when you look at the use of pollen and spores in solving crimes, again, what that's going to do is it's going to allow you to know where the individual, whether you're talking victim or whether you're talking potential suspect, where they may have been based on the amount of pollen that they have and the types of pollen that they're going to have. So that'll allow you to be able to know not only the season that they're there, but then also the region that you would find those particular plants flowering or producing reproductive structures at that particular time of year. So that's very, very useful. Now, obviously, to collect pollen or spore evidence, you have to be extremely careful because pollen and spores um, are so common. It's very easy for any of us to come in contact with them. And so again, if we're talking about Locard's exchange principle, the idea that every contact is going to leave some type of trace, just you and I going through our daily lives creates the opportunity for potential spore collection, not deliberate but unintentional. And so, if we were then to be analyzing evidence and maybe we didn't take precautions to cover up our clothes or whatever, some of that pollen or some of those spores could be deposited on the evidence and obviously could be very misleading in terms of what they show. So something to be mindful of there. And so again, the whole cross contamination thing, much like we talked about with hair, much like we obviously talked about with fibers and textiles, still holds true when we're talking about pollen and spores. Locard was well ahead of his time in terms of his understanding of how material gets transferred, and obviously that's going to be the same when we're talking about pollen and spores as well. So pollen and spores can collect in a number of locations, whether you're talking about humans in terms of our hair, on our clothing, on tire treads, in our nasal passages, right? Even in our lungs or lots of different locations, obviously, that we can collect this material. But if you're talking about other organisms as well on their fur, feathers, if you're talking about on insects, could be on different regions of their body, things like that. So these materials can find their way into lots of different locations, and so again, really our challenge as a scientist would be to figure out, okay, where are these things at, and are they of significant value? All right, so do they hold value from a forensic standpoint because of maybe the type or what they are? Okay. So when we talk about collecting samples of pollen and spores, you want to be able to have not only your evidentiary material, so stuff that's either collected from the victim or from um, maybe from a car or from some article of clothing or whatever it might be, or maybe it's from the suspect. But you want to have known control samples as well. So if you have a region where a particular body was located, you might call a palynologist and ask them to come out and collect samples so that you can determine whether or not the samples that are on the individual match what's in that area at that time period. Again, you could use that then to determine maybe length of time that they've been a body has been dumped or amount of time since someone has been in a particular location. There's lots of ways that it could be used. So the control samples, they're going to build that pollen fingerprint, if you will, that we talked about previously as a comparator. So again, you're going to wear gloves, maybe even moon suits, though not necessary most of the time, but definitely gloves. They're going to use paint brushes, the very small paint brushes, and then lifting tape. The important thing to do, obviously, in collection of these things, you're going to either have to use different paint brushes for each collection, or they're going to have to be thoroughly cleaned before you use them to collect subsequent samples. Most of the time, I would anticipate they're going to probably be one-off uses, so you'll use it and then simply dispose of it, or select a different to use to collect your next sample. Once the materials have been collected, they get put into their own individual containers. Again, they're going to be labeled with time and date, who collected them, how it was collected, so on and so forth, following all the normal um, chain of custody type of stuff. Excuse me, because again, you need to be able to establish that sequence that's been secure, so that information can be presented then in a court potentially. In addition to that, photographs of everything, much like we've talked about previously, you want to photograph everything as your documentation. Same is going to be true for this as well. The photographs are going to show not only what types of plants are present in that particular location at the time that you are there, but it's also going to provide evidence that shows how those samples were collected. So again, that just further strengthens that chain of custody, can be used or presented in a court to help explain kind of what process was taking place, so that you can ensure that uh, the technique was sound, the evidence was collected correctly, your exemplars or your control samples were collected in a way that protects their integrity and protects the integrity of the case as well. So that ends up being very, very useful. Samples put into sealed plastic bags, again, because most of this stuff, because of its durability, isn't going to degrade or decay very quickly, which is good. If you have vegetation that you've collected for whatever reason, maybe to show where the structures are located or whatever, they need to be first dried and then put into envelopes, paper envelopes, to preserve their integrity. And again, anything that falls off of those pieces of plant material then can be collected and used as further pieces of evidence to support the particular case, or to show uh, what was present in that location at that time period. In terms of analyzing the materials that end up being collected, uh, again, you want to try to do things that are going to be non-destructive. And so transmitted light or phase contrast microscopy, again, just using different forms of light and different filters, allows a scientist to be able to analyze what has been collected. Once they get drawings and sketches and photographs, obviously, of what's been collected, they're going to then compare those to reference books or journals or uh, uh, website searches, or any of those number of things. And once they think they have identified the plant that that particular material has come from, oftentimes they will then visit an herbarium to examine living plant material, collect samples to be able to make a positive identification, if that makes sense, to make sure that what they thought they were looking at is in fact accurate with what they saw. In the event that greater detail ends up being needed, you can always, or these scientists oftentimes will fall back on and use a scanning electron microscope, being able to see a much, much greater amount of detail with those scanning electron microscopes. The magnification is just astounding as compared to even the best light microscopes. So that ends up being beneficial there. But as I mentioned, palynology is going to be used in conjunction with other pieces or parts of the investigation, um, to aid in describing or explaining what took place at that particular time in that particular location. Okay. All right, so that is our section of notes here on pollen and spores. The next topic that we will be getting into will be that of soil. If you have any questions, make sure you reach out to me. Otherwise, I will talk to you soon.